Hi, it's Dr. Barr, and I love me some histology. We're going to talk about support cells of the central and peripheral nervous systems. By the end of the session, I want you to be able to recognize and distinguish the morphology and function of support cells of the central and peripheral nervous systems. Within the CNS environment, you have several cell populations. We talked about neurons in previous videos. With the image that's shown here, you have these large cells, which are the neurons, but you have numerous support cells, which are glial cells, and the nuclei that are located in the background. They support neuronal survival and activities. They're 10 times more abundant than neurons, and they develop from progenitor cells of embryonic neural plate. The neural pill, NP, which are your fibrous intercellular network of cells here, are comprised of neuronal and glial processes. Glial cells of the CNS and PNS include astrocytes, which provide physical and metabolic support, the oligodendrocytes, which produce myelin within the CNS. We talked about Schwann cells already in a previous lecture, and then ependymal cells, which produce and circulate CSF, which we've talked about in the meninges section. They line the ventricles and the central canal. Also microglial cells, which have similar function to macrophages. They are antigen-producing cells, and they have phagocytic properties. I mentioned before that Schwann cells are located in the peripheral nervous system, and they support myelinated and unmyelinated nerve fibers and produce myelin surrounding the axons. Satellite cells are analogous to Schwann cells, and they surround ganglion cell bodies. Please review this video on CNS histology.
The central nervous system, or CNS, consists of the cerebellum, cerebrum, brainstem, and spinal cord. The neuron is the basic working unit of the nervous system, and the neuroglia, or glial cells, are the non-neuronal cells that support and protect the nervous system. The central neuroglia includes astrocytes, oligodendrocytes, ependymal cells, and microglia. The peripheral neuroglia includes Schwann cells, satellite cells, and a number of cells associated with specific organs. Macroscopically, the CNS is made of white matter and gray matter. The difference in appearance is from the lipid-rich myelin sheaths that cover the axons present in white matter, whereas the gray matter consists mostly of neuron cell bodies, dendrites, astrocytes, and microglial cells. In this high-power image of white matter from the spinal cord, the axons are surrounded by clear white space, which is where the myelin was present before the tissue was processed to create the slide. The outermost portion of the cerebrum and cerebellum consists of gray matter, with their white matter present mainly in the deeper regions of the brain. On the other hand, the spinal cord has the opposite arrangement, with white matter mainly in the periphery and gray matter mostly located closer to the center, forming an H or butterfly-shaped appearance when looking at a cross-section of the spinal cord. In this low-power image, we can see the spinal cord's two posterior or dorsal horns closer to the top of the image, and two anterior or ventral horns at the bottom. The dorsal horns contain mostly sensory neurons, and the anterior horns contain mostly upper motor neurons. A simple way of remembering the location of the motor neurons is to remember that most cars also have their motors in the front or anterior part of the car. The entire CNS is also covered by layers of connective tissue called the meninges. The portion of the meninges that can be seen in this low-power image is the dura mater, which is the thickest and outermost layer of the meninges. The ventricles of the brain and the central canal of the spinal cord are filled with cerebrospinal fluid, or CSF, and have a lining of cuboidal or columnar cells called ependymal cells. These cells are responsible for producing CSF. If we take a closer look at the cells, we can also faintly see that the ependymal cells can have cilia, which help circulate the CSF within the central canal. The ependymal cells can also have microvilli, but they're typically too small to be seen with light microscopy. Now, let's take a closer look at the border between the spinal cord's white matter and gray matter. On the left is the white matter, where we can identify many of the purple-stained axons by the myelin sheaths that leave a white space that surrounds each axon. On the right of this image is the gray matter. Although neuron cell bodies can vary a lot in overall appearance, they're still usually recognizable by their prominent nuclei with distinct nucleoli. They're also usually much larger than glial cells. The different glial cells can be difficult to identify and differentiate from each other when using an H&E stain, except for oligodendrocytes. Oligodendrocytes can be identified by their small, round, and condensed nuclei. Their cell bodies aren't easily seen because their cytoplasm is unstained since it contains a lot of Golgi complexes which don't absorb the stain. Oligodendrocytes have many processes that extend and wrap around axons to form the myelin sheath for axons within the CNS. The peripheral nervous system's myelin sheaths are produced by Schwann cells instead. The gray matter surrounding all the cells is called the neuropil, which consists of a dense network of fibrous cellular branches or processes from glial cells and neurons. In the CNS, the star-shaped astrocytes are the most abundant type of glial cell. They have many long processes that extend in all directions from their cell body. Many of the processes have enlarged terminal ends called foot processes, which surround capillaries as part of the blood-brain barrier. The foot processes help induce and maintain the formation of tight junctions between endothelial cells. Astrocytes also produce structural proteins called gliofibrillary acid proteins, or GFAPs. These proteins can be used as a unique marker for immunostaining to help visualize astrocytes in tissue samples. In this high-power image, the GFAP-specific staining makes it easier to see the astrocytes in brown, as well as the foot processes that surround capillaries, as we can see them near the top of this image. Microglia are small antigen-presenting cells of the CNS that are found in both white and gray matter. They have short branching processes that actively move and are the only glial cells that also migrate to different parts of the CNS, which helps them find and remove damaged tissue, as well as function as part of the CNS's immune response. Similar to astrocytes, microglia can be visualized more easily using techniques like immunostaining. Immunocytochemistry is a form of immunostaining where the cells are immunostained after they've been isolated and suspended in liquid, which is why the microglia in this image are the only cells present and they're also spaced out relatively evenly. The pyramidal cells are the most prominent neuron in the cerebral cortex. These efferent neurons integrate sensory information and initiate voluntary motor responses. They get their name from their large pyramid-shaped cell bodies that have an apex that points towards the cortical surface of the cortex at the top of this image. At the apex, there's a large branching dendrite that extends up toward the cortical surface. The sides of the cell have much smaller dendrites that branch out to the sides, and its single axon extends from the base of the cell to synapse with other cells in the cortex or even deeper into the white matter. Together, the direction of the axon and the large dendrite give pyramidal cells their appearance of being vertically arranged within the cerebral cortex. Moving to the cerebellum, at low power, the cortex has 3 layers seen with an H&E stain. The innermost layer that's stained dark pink is the white matter. The purple middle layer and outer light pink layer are both a part of the gray matter. The outer layer is the molecular layer, and the dark purple layer is the granular layer. If we zoom in closer to the border of these 2 layers, we can see that it actually has 3 layers because of a single layer of neurons called Purkinje cells that sit between the upper molecular layer and lower granular layer. Immunostaining was performed on this section of tissue in order to see the Purkinje cells more clearly by staining them dark brown. These neurons have very large cell bodies with a highly branched dendritic system that extends into the molecular layer and an axon that extends down through the granular layer, which is often hard to see even with immunostaining.
So on that note, I will leave you with a recap of some key CNS support cells, such as the oligodendrocytes, astrocytes, ependymal cells, and microglial cells. I know the video in osmosis talked about Purkinje cells and other cells, but we really just want to focus on these key cells right here. On that note, email me or contact me if you need any questions answered or clarifications, and I will see you at the next lecture. Thank you. Bye!